ethical, legal, and social issues in embryology, ethical issues in stem cell research and genetic modification of embryos. Embryology plays a significant role in the field of assisted reproductive technology, ART, and the biomedical sciences. However, it is also a topic surrounded by various ethical, legal, and social issues, particularly in relation to stem cell research and the genetic modification of embryos. As advancements in genetic engineering and reproductive medicine continue, they raise complex questions about human rights, moral considerations, and the potential societal impact. 1. Ethical issues in stem cell research. Stem cell research holds tremendous potential for regenerative medicine and disease treatment, but it also generates significant ethical concerns. The key ethical issues in stem cell research are often linked to the source of the stem cells, particularly embryonic stem cells. Key ethical concerns: use of human embryos. Embryonic stem cells are derived from human embryos, typically from excess embryos created during in vitro fertilization, AVF. The ethical issue arises from the fact that in order to obtain these stem cells, the embryo must be destroyed, which some consider a form of destruction of potential life. Opponents argue that it is unethical to create and destroy human embryos for research purposes, as they believe human life begins at conception. Proponents argue that the potential benefits, such as curing diseases or regenerating tissues, outweigh the moral concerns about the destruction of embryos. Alternative sources of stem cells. The ethical concerns related to the use of embryonic stem cells have led to research into adult stem cells, which can be obtained from tissues like bone marrow or fat. Induced pluripotent stem cells, iPSCs, are another alternative, where somatic cells are reprogrammed to become pluripotent without using embryos. While iPSCs and adult stem cells eliminate the ethical dilemma of embryo destruction, they may not offer the same range of developmental potential as embryonic stem cells. Therapeutic cloning. Somatic cell nuclear transfer, SCNT, or therapeutic cloning involves creating an embryo by transferring the nucleus of a somatic cell into an egg cell. This process could produce stem cells genetically identical to the donor, potentially for use in treating diseases. Ethical concerns include whether it is morally acceptable to clone embryos for therapeutic purposes, as well as concerns about the possible commodification or exploitation of human embryos. 2. Ethical issues in genetic modification of embryos. The genetic modification of embryos, often referred to as germline editing, has become one of the most debated topics in modern embryology and genetics. Techniques like CRISPR-Cas9 have made it possible to alter the genes of human embryos, raising concerns about the potential benefits and dangers of such interventions. Key ethical concerns. Designer babies. One of the primary ethical concerns in genetic modification is the possibility of creating designer babies, embryos that have been genetically engineered to possess certain traits, e.g., intelligence, physical appearance, disease resistance. Critics argue that genetic modification could lead to eugenics, where certain traits are artificially selected and others are discarded, exacerbating societal inequalities and discriminating against those who are born with genetic conditions or without the preferred traits. There are concerns about the social pressure to conform to certain genetic ideals, which could lead to reduced diversity and increased discrimination based on genetic makeup. Germline editing versus somatic cell editing. Germline editing refers to modifications made to the genes of an embryo, which will be passed on to future generations. This raises concerns about unintended consequences and the long-term impact on the human gene pool. Somatic cell editing, on the other hand, involves altering the genes of non-reproductive cells, e.g., to treat disease, and does not pass changes on to future generations. Many scientists argue that somatic cell editing should be prioritized over germline editing due to the risks associated with permanent changes to the human genome. Unintended consequences and off-target effects. Gene editing technologies such as CRISPR-Cas9 can sometimes cause unintended changes in the DNA known as off-target effects, which could lead to unforeseen genetic mutations or health problems in future generations. Ethical concerns include the lack of certainty about the long-term safety and efficacy of these technologies, particularly when applied to human embryos. Human enhancement and inequality. There is concern that genetic modifications could be used to enhance human capabilities, such as intelligence, athleticism, or physical appearance, leading to a divide between genetically enhanced individuals and those who cannot afford such interventions. This raises social justice concerns and the potential for a genetic class divide, where wealthier individuals may have access to genetic enhancements, exacerbating existing inequalities. Third, legal issues in embryology and genetic modification. As embryology and genetic engineering technologies continue to evolve, legal systems around the world are grappling with how to regulate and govern their use. Key legal concerns. Regulation of gene editing. The legal framework for regulating germline editing is still in its infancy. In many countries, germline gene editing is banned or severely restricted, while somatic gene editing for therapeutic purposes is often allowed under specific regulations. There are international agreements such as the Oviedo Convention, Council of Europe, that attempt to establish common standards for the protection of human dignity, including restrictions on certain types of genetic modification. Intellectual property and patents. The patenting of genetic modification technologies, such as CRISPR-Cas9, raises concerns about intellectual property ownership and whether scientists, companies, or governments should have the right to patent technologies that could be used to alter the human genome. Legal issues also arise regarding ownership of genetic material, particularly in cases where genetic material from embryos is used for research purposes or stored for future use. Reproductive rights. Ethical and legal debates also extend to the right to reproductive choice. Some argue that genetic modifications, especially germline editing, could interfere with individuals' reproductive autonomy and the right to make decisions about the genetic makeup of their offspring. International disparities. Legal regulation of gene editing and stem cell research is inconsistent worldwide. Some countries have adopted strict laws to prevent certain procedures, while others have embraced more permissive policies. This discrepancy has led to concerns about genetic tourism, where individuals travel to countries with less stringent regulations to undergo controversial treatments. Fourth, social issues in embryology and genetic engineering. The use of stem cells and genetic modification of embryos can have broad societal implications, including concerns about accessibility, social inequality, and moral values. Key social concerns: accessibility of advanced technologies. Access to stem cell therapies and genetic modifications is often limited by financial resources, which may create disparities between wealthier and poorer populations. This could exacerbate health inequalities. As these technologies become more advanced and expensive, they may be available only to the elite, creating a genetic divide between different socioeconomic groups. Cultural and religious considerations. Different cultures and religions have varying perspectives on embryo manipulation, genetic engineering, and stem cell research. For example, some may believe that altering the genetic code of embryos violates divine or natural laws, while others see it as a potential means to improve health outcomes. Religious objections may be particularly strong when it comes to the destruction of embryos for stem cell research or the modification of human genes. Impact on future generations. The genetic modification of embryos can have long-lasting effects, as changes made to the genome will be passed down to future generations. This raises the question of whether society is prepared to make decisions on behalf of future generations and whether the risks are worth the potential benefits. 5. Conclusion. The ethical, legal, and social issues in embryology, particularly regarding stem cell research and genetic modification of embryos, are vast and complex. As advancements continue in gene editing and regenerative medicine, there is a need for clear ethical guidelines, comprehensive legal frameworks, and public dialogue to address these concerns. Striking a balance between scientific progress and human rights is essential to ensure that these technologies are used responsibly and for the benefit of society as a whole.